Hello, everyone. I'm very happy that we can learn together again today. Today, we are going to introduce Immunoblot. Immunoblot, often referred to as Western blot, is a popular and valuable method used in the fields of molecular biology, biochemistry, and immunogenetics. The name Western blot comes from its original historic race to achieve scientific results. Back to that time, three independent scientific teams were working on establishing the immunoblot method simultaneously. The team on the west coast of the United States named the technique Western blot based on their location. Although their research fell behind later, the interesting name persisted. This process enables researchers to visualize and analyze proteins of interest, revealing valuable information about their presence, quantity, and modifications. This technique has a variety of important applications, such as identifying and quantifying specific proteins, examining post-translational modifications, investigating protein-protein interactions, and comparing protein expression across multiple samples. Through these applications, Western blot enables scientists to better understand the complex processes occurring within cells, tissues, and organisms. So what is the principle behind this? Immunoblot, an insightful technique applied in scientific research, brings together the concepts of gel electrophoresis and immunoassay. By utilizing specific antibodies that have a strong affinity for the target proteins, this method allows researchers to identify and quantify proteins with great reliability. Next, let's talk about the main steps of the Immunoblot. This technique includes 5 steps. First is sample preparation. Cells or tissues are lysed in a cell lysis buffer using a high-speed homogenizer or ultrasound homogenizer to release cytoplasmic proteins by breaking the cell membranes and organelles. The sample is then centrifuged to remove debris, and the protein concentration in the supernatant is measured using colorimetric, BCA, or Bradford methods. Second is gel electrophoresis. The proteins in the sample are separated by size using polyacrylamide gel electrophoresis, or PAGE, under denaturing conditions, typically using sodium dodecyl sulfate, or SDS. Polyacrylamide gel is a cross-linked polymer composed of different ratios of acrylamide and cross-linker. During electrophoresis, the electric field causes the charged proteins to move towards the electrodes. The size of the proteins restricts their movements through the gel pores, causing larger proteins to move more slowly and smaller proteins to move more quickly. The denaturing conditions provided by SDS help to remove the protein structure, allowing it to move through the gel only based on its charge. This step helps to create a clear protein fingerprint based on protein size. Third is the transfer step. The separated proteins are transferred from the gel to a solid support membrane, typically made of nitrocellulose or polyvinylidene difluoride, or PVDF. This process is called electroblot. Transferring the proteins to the membrane allows for easier handling, probing, or antibody detection of the proteins. It also avoids the potential effects of the gel on downstream analysis. Additionally, the membrane properties allow for better interaction between the proteins and probes or antibodies, improving sensitivity and specificity. The transfer involves stacking filter paper, gel, membrane, and blot pad in an electrotransfer apparatus and applying a constant current or voltage for 1 to 2 hours, depending on protein size and gel thickness. After transfer, a membrane with a copy of the gel is obtained, which is used for subsequent immunoblot experiments. Fourth is blocking. The membrane is incubated in a blocking solution to prevent non-specific binding of the antibodies in the next step. Antibodies can bind non-specifically to gaps on the membrane where the target protein is not present, leading to background noise. To avoid this, the membrane is incubated in a blocking solution containing a protein, such as a non-fat dry milk or bovine serum albumin, or BSA, which occupies the gaps on the membrane without affecting the proteins. Fifth is antibody probing. The membrane is incubated with primary antibodies that specifically recognize and bind the target protein, typically overnight or for several hours at 4 degrees Celsius. Then, the membrane is washed several times to remove unbound or weakly bound primary antibodies, reducing background noise and improving detection specificity. A secondary antibody conjugated with an enzyme, typically horseradish peroxidase or HRP, is added to the membrane. This antibody recognizes and binds the primary antibody, generating a detectable signal. The membrane is then washed again to remove unbound or weakly bound secondary antibodies. 6 is detection. A colorimetric substrate is added to the membrane. The substrate is catalyzed by HRP, generating a visible color. Therefore, only the HRP conjugated secondary antibodies that are specifically bound to the target protein on the membrane will react with the colorimetric substrate and produce a visible color. This color reaction produces one or more distinct protein bands on the membrane, corresponding to the location of the target protein on the gel. By comparing the intensity or size of protein bands from different samples, researchers can perform quantitative or qualitative analysis of protein expression or post-translational modifications. Do you know the advantages of Immunoblot? First is high specificity. It can accurately identify and detect target proteins by using specific antibodies against the target protein. Second is detectable protein modifications. It can detect post-translational modifications of proteins such as phosphorylation and acetylation, which often affect protein function and activity. 3 is high sensitivity. Immunoblot has high detection sensitivity and can detect very low abundance target proteins in samples. 4 is that it's combinable with other techniques. Immunoblot can be combined with other biological experimental techniques such as immunoprecipitation and mass spectrometry to further study the function and interaction of target proteins. 5 is a relatively low cost. Compared to other protein detection techniques, the required experimental equipment and reagents are relatively inexpensive. However, there are also apparent disadvantages, such as relatively complicated experimental operations and possible non-specific signals. Nevertheless, these issues can be effectively resolved by optimizing experimental conditions and using high-quality antibodies. Overall, Immunoblot is a valuable protein detection and analysis technique. In clinical diagnosis, Immunoblot is usually not the first choice, but serves as a complement to other screening methods. For example, in HIV testing, HIV antibodies in the serum can be screened using enzyme-linked immunoabsorbent assay, or ELISA, but positive results need to be confirmed by Immunoblot. The same applies to the detection of Lyme disease, hepatitis C virus, or HCV, and in autoimmune diseases. This is because Immunoblot is labor-intensive and time-consuming, making it unsuitable for large-scale screening.